Metabolic refers to the biochemical processes that occur within living organisms to maintain life. These processes involve the conversion of nutrients into energy and the synthesis of molecules necessary for cellular function and growth. Metabolism encompasses various interconnected pathways, including the breakdown of molecules, catabolism, to release energy, and the synthesis of molecules, anabolism, to build cellular components. In the context of cancer, metabolic alterations refer to changes in these pathways that allow cancer cells to sustain their growth and proliferation. This can include shifts in energy production, changes in nutrient utilization, and adaptations to environmental conditions to support the unique demands of cancer cells. Understanding the metabolic characteristics of cancer is crucial for developing targeted therapies that exploit these vulnerabilities while minimizing harm to healthy cells. How changing our metabolism we can avert cancer. Changing metabolism to target cancer involves exploiting the metabolic vulnerabilities unique to cancer cells while minimizing adverse effects on healthy cells. Several strategies have been explored. Targeting aerobic glycolysis. Cancer cells often rely on aerobic glycolysis for energy, even in the presence of oxygen. Therapies aimed at inhibiting enzymes involved in glycolysis, such as hexokinase or lactate dehydrogenase, can disrupt cancer cell metabolism and induce cell death. Inhibiting nutrient uptake. Cancer cells have increased nutrient demands to support their rapid growth. Drugs targeting nutrient transporters or metabolic pathways involved in nutrient uptake, such as glucose or glutamine, can starve cancer cells of essential nutrients, inhibiting their growth. Modulating mitochondrial function. Mitochondria play a crucial role in cellular metabolism and energy production. Targeting mitochondrial function, such as inhibiting mitochondrial respiration or promoting mitochondrial dysfunction specifically in cancer cells, can induce cell death selectively in cancer cells. Exploiting synthetic lethality. Synthetic lethality involves targeting combinations of genetic mutations or metabolic pathways that are lethal when disrupted together but not individually. Identifying synthetic lethal interactions specific to cancer cells allows for targeted therapies that selectively kill cancer cells while sparing healthy ones. Immunometabolism. Manipulating the metabolic environment of the tumor microenvironment can influence immune cell function. Enhancing the metabolic fitness of immune cells, such as T cells, through metabolic reprogramming or nutrient supplementation, can improve their ability to recognize and eliminate cancer cells. Metabolic drugs. Developing small molecule inhibitors or metabolic drugs that specifically target metabolic enzymes or pathways dysregulated in cancer cells can provide targeted therapies with fewer off-target effects. By understanding the metabolic alterations in cancer cells and developing strategies to exploit these vulnerabilities, it's possible to develop more effective and selective therapies that can overcome cancer while minimizing harm to healthy tissues. However, it's essential to continue research efforts to identify and validate promising metabolic targets and therapeutic approaches for clinical use.
With knowledge and proper nutrition, we can beat cancer in the cell. No chemo, no chemo, no chemo.